Welcome back after lunch. We're going to get started here with session number 2 of the conference. So today I have the opportunity to chair this session on the topic of behavioral pharmacology and abuse liability. And so what we've done is modified just a little bit the structure, so the presenters, so that they don't have to turn their heads, are going to stay in their seats until the Q&A, and then each will come back up. I'd like, so our first presenter today is going to be Dr. Charles France. And so Dr. France will be discussing preclinical assessment of abuse liability and therapeutic potential. And so Dr. France is the Robert A. Welch Distinguished University Chair in Chemistry and Professor in the Departments of Pharmacology and Psychiatry at the University of Texas Health Science Center at San Antonio. Dr. France has been continuously funded by the NIH for more than 35 years, has published more than 300 original research studies, chapters, and reviews, and has trained more than 100 trainees, which really interesting, running the gamut all the way from high school students to postdoctoral fellows. So really excited to hear the talk, and I will turn the floor over to you, Dr. France.
Thank you. Let me figure out how to get the slides on. That wasn't it. There we go. Okay, good. Thank you. Thanks to Steve for inviting me to such a beautiful place in the world to listen to such interesting science. I've heard for many years about this meeting and often wanted to get up here. What a beautiful place. And also thanks to Chris for making the very comfortable arrangements to get here and be part of this meeting. At the very end, about a molecule for which I have to make the disclosure. It's on this screen. I could talk more about that later if you like. There's 2 parts to my talk. The first is to talk about the preclinical abuse liability assessment. That's mostly of new chemical entities. As Jack said earlier in the day, anything that really acts in the CNS and is going to be developed probably is going to require an abuse liability study in animals and possibly in humans. In the latter part of my talk, I will discuss a little bit about therapeutic potential. These are very closely related topics, as you'll see, because they use generally the same procedures, ask very much the same kinds of questions. This kind of work in non-human species to evaluate abuse liability. This document defines drug abuse as the intentional non-therapeutic use of a drug to achieve psychological or physiological effect. And it's worth noting that these kinds of experiments or studies fall under safety pharmacology, not under toxicology. That's important because it determines the kinds of doses that you study. In pharmacology, toxicology doses studies, you run the doses up to the top and probably to mortality. That's not at all the case in these kinds of experiments. And they delineate 4 general kinds of experiments that might be used. General behavioral studies. I'm not going to have time to talk about those today. And the heart of it really is drug discrimination and self-administration, or in some cases, condition place preference. They also talk about physical dependence. I won't have time for that today. The second document that has guided this work, because a lot of clients who've come to us have come from the European community, is from the European Medication Agency, and the title of their document is Guidelines on the Non-Clinical Investigation of the Dependence Potential of Medicinal Products. Not to be confused, they're not talking specifically about physical dependence as we might think of it, but instead their definition of dependence is propensity to give rise to need for repeated doses to feel good or avoid feeling bad. And again, they talk about general behavioral procedures that could be used. Note the overlap between these 2 documents, both talking about potential, that is, what's the predictive validity of these studies in non-human species, and both of them talking about generally the same sorts of behavioral procedures. I'll talk today about drug discrimination and self-administration. This is the drug discovery and development pipeline. I'll apologize in advance. Maybe everybody in the room knows what I'm going to talk about today, because I know there are people in here who have done abuse liability studies, so I'll apologize in advance. But this goes from early drug development up through post-marketing. And the kinds of studies I'm talking to you about today most commonly occur in this middle area of clinical development after phase 2 clinical trials have been completed. That's important because it's only at that point in time that you know the therapeutic doses that are likely to be used in humans, and therefore you can estimate the kinds of doses that are going to be relevant in animal studies. Prior to that point, you're really shooting in the dark on doses. That said, having done many of these studies over the last 30, 40 years of my career, we have done them much, much earlier in development. We've done them way pre-IND, particularly when companies thought that they had a drug which was a potential problem, either because of its target or its chemical structure. They wanted to pop it into animals quickly to see what was happening. It's a very cost-effective way to preclude spending tens of millions of dollars down the road to find out something you could have found out earlier. So that's when we generally do those studies. And of course, the goal here is an NDA. Jack talked about that this morning. That's a big, big package of data. And in that package of data are some important non-clinical sets of data. One is pharmacokinetics. There's a lot to that. I'm not going to talk about it today other than to say once one has those pharmacokinetic data in animals, it's much easier to design and do reasonable abuse-related studies in those non-human species because you know the time of peak effect, duration of action, et cetera, et cetera. A number of things have to be considered for these sorts of studies in non-human species. Rats are the preferred species by many, many agencies, and certainly by the FDA. They explicitly state that rats should be used, although there are cases where other species are more suitable. And it may be because the metabolic profile does not align well between rats and humans. We certainly have had instances of that. And there may be other cases where there are pharmacological factors that would preclude the use of rats and warrant it in other species. As I said, it's not toxicology, it's safety pharmacology, and as a consequence, we tend to study up to 2, 3, maybe 4 times the expected clinical exposure in animals, and it's important that you test it at Tmax. You want to be sure that you maximize the possibility of detecting a positive signal if there is a signal there. By the FDA's authority, we must have a positive control to demonstrate assay sensitivity. Hopefully that's from the same pharmacological class as the drug that's being studied, and otherwise the same indication. So these behavioral studies pose 4 general questions. First of all, is it safe? Things like Irwin studies that do run doses up to very high doses. I'm not going to talk about those today. Is the new chemical entity similar to a drug of abuse? That is it qualitatively similar? And for that, drug discrimination is exquisitely well suited to answer that question. Does it have abuse-related effect? Typically, that's self-administration. It can be condition-place preference. And then does it produce physical dependence? And that's only for those appropriate conditions where drugs are going to be given under conditions where physical dependence might be warranted. If it's a once-a-month medication, that's not likely going to be part of the package. So first I'll talk about drug discrimination. Again, probably everybody in here knows a little bit about that, but I'll quickly review the procedure. You have to pick the appropriate drug, and that's probably based upon the target of the new chemical entity. The most responsive species, I put responsive in quotes because that's directly from the FDA document. Most often that's rats. And they suggest a simple schedule, FR10, train the drug, test a vehicle, have a positive control, then do the test compound. I can tell you, having done this for many years, we do lots of variations on a theme. There are FDA guidelines, but they are only guidelines, and in some situations, other kinds of conditions are warranted. Just as you press the accelerator when you see a green light and you press the brake when you see a red light, this is a detection experiment that I'm talking about. In this case, it's a visual detection. In the case of drug discrimination, it's an interoceptive stimulus detection. Presumably the animal tells us whether or not they receive the drug. Typical experiment, an animal's in a chamber with 2 levers. They get an injection of drug prior to that session, and if they respond, in this case on the left lever, they get a reinforcer. If they respond on the right lever, they get nada. On other days, they get a vehicle injection prior to the session. The previously reinforced left lever is no longer active, and now they can respond on the right lever to get a reinforcer. And if you do this right with the appropriate dose, animals become incredibly effective at telling you whether or not you've given them the drug by choosing the left lever over the right lever. You then can ask them questions. You can give them the training drug, new drugs, combinations of drugs, all sorts of things, and say, hey, does this make you feel like the drug to which you've been trained? And the thing about drug discrimination for those who are not aficionados is it's very pharmacologically specific. Animals don't learn drug versus my drug versus all other drugs. They learn a specific aspect of a drug versus everything else in the universe. And that makes it both a very useful and in some cases restricted procedure. So just like red light, green light, the animals learn to detect the drug. One kind of experiment that's done with drug discrimination is when you know the target and it's easy to pick the reference compound. And I'm going to give you an example of that right now. Increasingly, I should say, drug discrimination, for our perspective, is becoming an important part of the package because we're studying all sorts of novel drugs that are not particularly good reinforcers in self-administration experiments. So drug discrimination becomes comparatively more important. This is one for drug we're studying. It's a psychedelic, the serotonin receptor agonist the company was trying to develop. And this is a discrimination with rats. In the upper panel is presented the percentage of responses on the drug lever. So if it's 100%, they're responding 100% on the drug lever. If it's 0, they're responding on the vehicle lever. The bottom simply shows the rate at which they're pressing the lever. And you can see here that this training dose of DOM, a well-known psychedelic, 0.56 milligrams per kilogram, when rats get that, they respond on the drug lever. They're well trained. Here's the negative control vehicle. They respond predominantly on the vehicle lever, just like the red light, green light. And there's a dose-related relationship there. And that's one nice thing about these sorts of experiments is they're really, really very quantitative and very precise. Need a positive control. The one that was selected for this experiment is DPT. It's another well-known psychedelic drug, and it produces complete drug-appropriate responding. And in this case, the not-to-be-disclosed test compound did not produce significant DOM lever responding up to doses that markedly decreased responding. So the failure of this drug to produce DOM-like discriminative stimulus effects would predict that it's not likely to have DOM-like or psychedelic-like effects in humans. Not guaranteed, but that's the way that the procedure is used. There are other situations increasingly these days where you don't have any idea what the training drug should be, and you just have a new chemical entity and they say, do a drug discrimination experiment. You say, well, what should I do? Well, one thing you can do, and I should say we've jumped through all of these hoops, is to train a batch of known drugs of abuse. So train different groups of animals to discriminate different kinds of drugs, maybe an opioid, maybe a stimulant, maybe caffeine, maybe alcohol, and then put the test compound into those assays and hope that you get a hit. But if you get a hit, you might not learn much. And if you don't get a hit, you don't learn anything. In the meantime, you have spent a lot of time and energy training a lot of different animals. A second one is to take the new chemical entity and actually train it. And I should say we've done that too by recommendations of clients and apparently the FDA, though I don't understand that, because it provides a really complicated experiment. First of all, you have to be able to train something you don't know very much about. So you have to pick the dose, the route, and the pretreatment time. And once you get it trained, then what test compounds do you put in? Every known drug of abuse, or do you select this one or that one? Again, it turns out to be a little bit of a crapshoot. And if you get a hit, you might learn something interesting. But if you don't get a hit, again, you would have wasted a lot of energy. My own experience, if it's not A, that is, if you don't have a known target that you can train and test your compound against, you're going to burn a lot of rubber with options B and C. We haven't seen that bear much fruit. Next, I'll talk about drug or self-administration, which really has been the gold standard of abuse liability assessments for many, many years. And I'll tell you why that's the case. The self-administration procedure, again, apologize for those who know all of this, as animals typically press a bar to get an intravenous infusion of drug. It's nice to have a reference compound from the same pharmacological class, though it's not required because animals are promiscuous. They self-administered lots of different drugs. And although there are cases where history of self-administering a particular drug impact a subsequent test, there are many, many cases where that's not the case. Vehicle from the FDA's point of view is the only appropriate control here. That is an inert substance. Typically, we test a reference substance, the negative control, the test substance, and then reconfirm the assay sensitivity with the reference substance. I'll show you that. Self-administration is critically dependent upon pharmacokinetic profile. That's a plus and a minus. If drugs do not have a rapid onset, they're not likely to be effective reinforcers. And that can limit the effectiveness of the assay in some cases. The FDA says in FR10, as I said earlier, we've done lots of different schedules of reinforcement. The European guidance actually indicates progressive ratios are useful under some situations. It's a particularly useful assay because of its predictive validity. It's not 100%, but it sure as heck is very strong. This shows lists of drugs that are very well self-administered by animals on the left hand, drugs on the right hand, drugs not so much. Now you're looking at a guy who believes you can train monkeys to play the piano, and I think you can. And you can probably train them to take any drug in the pharmacopoeia if you're willing to work hard enough to do that. Steve Goldberg was good at that. But in terms of a good, robust signal, these are the ones that we tend to choose. So it's the gold standard. Animals press a lever, they get an infusion. These are the kinds of drugs that are often the reference substances. I'm going to give you an example that doesn't use any of these drugs. we were recruited to do the studies on cannabidiol or CBD that the FDA required for the abuse liability assessment. And cannabidiol, though being a cannabinoid, doesn't have an obvious reference substance because delta-9 THC, the active ingredient, is not a very robust reinforcer in animals. You can get it, but it's weak. So in negotiations with the FDA and the client who came to us, they decided, well, CBD has sedative effects, have a sedative drug as the positive control. And they decided on benzodiazepines. Well, that seems reasonable, but rats don't take benzodiazepines very well. So this study had to be done in monkeys. So monkeys pressed the lever to get midazolam, then we tested vehicle, then CBD, and then reconfirmed with midazolam. These are the actual data. The ordinate shows the number of infusions monkeys take in a 90-minute session, responding under an FR30 schedule. They take about 20. The negative control, you put vehicle in, they pretty much lose interest in pressing the lever. We tested several doses of CBD. No indication of any positive signal, not unexpectedly. Because the animals had gone for an extended period of non-reinforcement, we wanted to reaffirm the sensitivity of the assay, put midazolam back in, boom, they took it very nicely, went back and had to study bigger doses of CBD. We went up here, I think, to 4 or 5 times the multiple of the expected clinical exposure in humans. This drug, Epidiolex, is used to treat treatment-resistant seizures in kids especially, and then reconfirming with midazolam at the end. So this is a classic preclinical abuse liability study. Positive control, test the drug, negative control, reconfirm the drug at the end. And we've done lots and lots of these. This is a quirky one because of midazolam and CBD. Epidiolex was scheduled initially in 2018 as a Schedule 5. It became unscheduled in 2020. I can share the data with you because of all the studies we've done for companies, I can count on one hand how many times we've been able to disclose the data. This is one of them. The challenges to self-administration are they are critically dependent on PK profile. You can get false negatives here. If drugs have a slow onset of action, pro-drug metabolized to something active, you can miss it with a self-administration experiment. So it's only part of a package. How to pick the reference drug. That used to be easy. It's less easy now, as evidenced by the CBD midazolam example that I just gave you. Not all abuse substances are self-administered. Again, you can teach a monkey to play a piano, but I think in terms of a robust signal, there are some drugs that are going to have abuse liability that aren't going to be picked up here. The last part of my talk, I'll discuss a little bit using similar procedures to look at the therapeutic potential of new chemical entities, so drugs that might be used to treat substance use disorders. Generally asking the question, does it block discriminative stimulus effects? Does it block the self-administration? Does it block reinstatement? That now comes into play. And does it block the development of physical dependence or expression of withdrawal? I won't have time to talk about the last one, but I'll talk about the first 3. The example I'm going to give you is lorcaserin. It's a really complicated molecule. If you don't know it, it has lots of different actions, but one of them is it's an agonist at serotonin 2C receptors, and it was marketed because it reduces appetite in humans. And there had been a growing body of evidence, Catherine Cunningham and others, looking at serotonin 2C receptor agonists that indicated, well, these drugs might be usable for treating stimulant use disorder. This drug was approved 2012 by the FDA, so it was available for humans. And we did a series of experiments on it as a potential treatment for cocaine use disorder. The first thing we looked at was cocaine discrimination. These are monkeys that discriminate cocaine, as I showed you earlier with DOM, dose-related switch in responding from the vehicle to the drug lever. Thankfully, lorcaserin did not produce cocaine-like discriminative stimulus effects. It'd be tough to develop a medication if it was cocaine-like in a discrimination procedure. And when combined with cocaine, it had mixed effects. In monkey MU left and LA right, yes, it did shift the dose response curve right into the down. That's encouraging. The monkey in the middle, DU, not so much encouraging, but there was some hint of a signal here in modifying discriminative effects of cocaine. So a qualified yes. In terms of reinforcing effects, this shows dose response curves for lorcaserin in decreasing both responding for food, that's the open symbols, and responding for cocaine, that's the closed symbols. And yes, it decreases both, maybe not surprisingly, since it was marketed to reduce appetite. But there is some hint of selectivity at 3.2 milligrams per kilogram, enough for those who were supporting this study to say, pursue it further. So we did. One thing you want to do is look at repeated dosing with the drug. A medication needs to be effective not after one injection, but after a bunch of injections. So that's what this slide shows. Daily treatment with lorcaserin in monkeys self-administering cocaine, and it suppresses responding beautifully to less than 50% of control. That entire time, several weeks, stop the lorcaserin treatment, they recover beautifully. So that was a good positive signal. We wanted to know how robust that was, and back to Jack's comments, progressive ratio schedules can be useful for that. This is a progressive ratio dose response curve for cocaine. So each response requires more and more responses, each infusion. First one might cost you 30 responses, second one 60, third one 120, et cetera. And you can see with the largest unit dose of cocaine, 0.1 milligrams per kilogram, the monkeys are responding 700 or 800 times for a single infusion. It's a very powerful reinforcer. And yes, lorcaserin decreased progressive ratio responding quite nicely. It was another good, strong signal that it might be modifying the reinforcing effects of cocaine. So with regard to self-administration, lorcaserin was also quite effective under these conditions. There is increasing interest in reinstatement procedures in animals because it's thought to have a relationship to relapse in humans. And for these sorts of experiments, we do this kind of thing. This now shows on the ordinate the number of responses in a session, not the number of infusions, but the number of responses. And the first data point shows what happens when monkeys are actually pressing levers to get intravenous infusions of cocaine on an FR30 schedule. They make 700 or 800 responses. They're then put in the box, no cocaine, no cocaine-associated stimuli, no nothing. Just sit there, and they just sit there. We then expose them to the chamber again, and now if they press the lever, they get the lights that come on with cocaine infusion, so it's cocaine-paired stimuli. No cocaine is administered. And they respond pretty avidly, making 400 or 500 responses in a session. So this is the classic cue-induced reinstatement in monkeys. And if you add to that a single shot of cocaine before the session, you get a whole lot more reinstatement. So this is the classic model. The animals are in the box. They're not getting cocaine in the box, but they got a little squirt of it before the session. But if they respond, the lights come on and off. And you then say, well, okay, does a drug impact this reinstatement of responding? And yes, lorcaserin did. In a dose-related manner, it decreased reinstatement responding quite nicely down to levels that would be achieved with just the stimulus lights alone. So yeah, there was a clear signal here with lorcaserin. Story sounding good, right? There are strengths to the reinstatement procedure, insofar as it sort of looks in some ways like what happens in people. Drugs can reinitiate drug taking. Drug-associated stimuli can reinitiate drug taking, and stress, lots of things can reinitiate drug taking in humans, and they can do that in animals too. The weakness of the assay, though, is that its predictability is not terrific, and there are temporal mismatches. For humans, the longer they go without taking a drug, at least in some situations, the less likely they're going to relapse. In some of these animal models, the longer the animal is after having actually self-administered drug, the greater the reinstatement. So that's sort of a mismatch. Voluntary versus forced abstinence. The value, I guess, could be debated and probably shouldn't be in a bar, and you can tell which side of the argument I would be on. The good news story ends here because lorcaserin failed miserably in clinical trials with cocaine. We did other work with it in combination with other drugs, but it failed in clinical trials, so the plug was pulled. And in 2020, the FDA asked them to take it off the market because of increased cancer risk. So it was a good story to begin with, but not a happy ending. I'm going to give you one last example of a medication development, the sort of thing that we're doing in our laboratory. And this has to do with a drug called methocinomox, or MCAM. It's a fairly close relative to buprenorphine, discovered by John Lewis, same person who discovered buprenorphine. It's an opioid receptor antagonist. We're developing it avidly. I had a meeting on it this morning. We hope to have it in phase 1 within about 8 weeks if we get our IND approved. These are monkeys self-administering fentanyl. They take about 20 infusions in a 90-minute session. If you give them a single shot of MCAM, it goes down to the point at which they would self-administer if there was just saline in the syringe. That single dose of MCAM suppresses, or at least decreases, fentanyl self-administration for several weeks. In fact, it's not until 1 month after that injection that this group of animals recover their sensitivity to fentanyl. That's particularly interesting because MCAM can no longer be detected in blood after about 6 hours. So it's a PK and not a PD effect. We think we know why it's happening, but we're not sure. It's not because the monkeys can't respond, because on intervening days where there's the break in the blue line, we replace the fentanyl with cocaine. They took cocaine beautifully. You wouldn't expect MCAM, an opioid receptor, to block cocaine, and it did not. Okay, I've told you about basically drug discrimination. I hope I didn't speak too slowly, and self-administration, and how these things are used in 2 venues: to look at the abuse potential of new chemical entities, but secondly, to look at things that might be effective therapeutics in the long haul. Behavioral pharmacology has been fantastically important in this regard over the last 50 years. It's highly quantitative. You can study a very broad range of conditions, things you can't do in people, high predictive validity. You can do this early in development and save tens and tens of millions of dollars if you pick up a signal, so it can be very cost effective. Early detection of abuse potential. You can get false positives. As Jack said this morning, he thinks rats are more predictive than humans. I think that's a nice thought. Maybe monkeys are too. Clinical data historically have always trumped, I apologize for the use of the term, non-clinical data. And I think maybe that situation is beginning to change as we go forward in the future. We get more novel chemical entities. In terms of the preclinical therapeutic potential assessment, I would have to say these procedures have probably been useful, but what have we developed? Not much. So is that because we're not getting the right kind of drugs? We don't have the right behavioral procedures. We're not asking the right questions of animals. Or, my view, we're expecting magic bullets. And in fact, no one would expect a single molecule to cure such a complicated disease. But I think the behavioral procedures are still useful in the grander scheme of things. I would have to mention impulsivity here, both because of Warren and his colleagues. And I should say there have been a number of occasions where clients have asked us to look at measures of impulsivity with medications they were developing for substance use disorders. In most cases, it was for stimulant use disorders. And in some cases, they were studying stimulant-like drugs, and I'm supposed to be quiet, and I will. And they were concerned about the fact that they may be increasing impulsivity, and the things we have used are delay discounting. Time is up. So I won't talk about that, but simply say thank you and thanks to people who've done the experiments. And later I'll take any questions that you might have. I like this schedule control behavior. That's very good. How do I turn that thing off? You try it.
Thank you, Dr. France. So our next presenter is going to be Dr. Rick Bevins, and he'll be presenting on learned amplification of nicotine use and misuse liability. So a little bit of background, Dr. Bevins is the Mildred Francis Thompson Professor and then Director of the Rural Drug Addiction Research Center at the University of Nebraska-Lincoln. Putting numbers to it, he says 1996. I say for 29 years, Dr. Bevins has led the Behavioral Neuropharmacology Lab at UNL, where his research explores why people start using drugs, what drives relapse, and how science can inform better treatments for substance use disorders. His work is supported by the NIH and state partners and focuses on substances including methamphetamine, cocaine, nicotine, opioids, and alcohol as a couple of examples. So I'll turn the floor over to you.
First, Charlie, that was an amazing talk. Thank you for that. And thanks for the morning speakers too as well. Thanks for the invitation. I'm just honored. This is such a, I came to the very first one of these, Steve. I don't know if you remember. And then I've been able to come back now repeatedly. And this is just one of the most amazing conferences. So kudos to you and your team. And thank you for the invitation. I have no conflicts of interest to report for this talk. It's really nice to be amongst colleagues and friends that recognize both the difficulty of nicotine addiction, its health harms, and the struggle of nicotine use disorder. I know in this day and age, it's surprising when I say that's not shared. Here's a review I received recently. With the advent of many nicotine replacement strategies and increased cost of cigarettes per se, nicotine use is already a pretty well-handled public health issue. Yeah, anonymous reviewers, right? So, I've often been asked, like, what if the reviewer was in the room? And I'm like, please, I really one day hope so. And so we've tried to piece together like what this reviewer was, who they are, and with AI tools and everything now, we have kind of an idea who it might be. I don't see this person here today though, so I don't think the reviewer's here. Nicotine is obviously addictive. It's obviously harmful. This is the 2025 report from the World Health Organization. Here's the Director General of the WHO saying, despite overwhelming evidence of tobacco's dangers, many users still don't know or underestimate its risk, and the powerful grip of nicotine addiction makes quitting extremely difficult. Meanwhile, tobacco companies exploit packaging to make their products appealing while downplaying their devastating health effects. course, addiction scientists working for the tobacco industry knew this a long time ago. We heard just an absolutely amazing historical tour to the modern relevance from Jack this morning. So thank you for that. But here's just two quotes that were unearthed in all the documents that had to be released with all the lawsuits. And, we are in a nicotine rather than a tobacco industry. No one has ever become a cigarette smoker by smoking cigarettes without nicotine, right? 1967-ish, 1971. Nicotine's addictive. But there's a puzzle to be solved here, because though it has a powerful grip, we also know as early as in the 70s and 80s, it was very difficult to get self-administration, as we heard. Here's Doherty, and he's lamenting about the reinforcing properties of nicotine are weak. He's talking about the primary reinforcing, and he goes through and talks about it's not dose effect sensitive, schedules of reinforcement. So all the classic things we just heard Charlie talk about are very important. Now, in fact, this is the case. So I'm just going to show off a little data from my recent PhD student who's now a postdoc at the University of Kentucky, Dr. McNeely. And this in the self-administration tasks. Thank you again, Charlie, for introducing that. We have a rat here, has jugular catheter that has been implanted. It has a choice between two responses. One response is the active response. We'll give it nicotine or saline in this case. And the other one is an inactive response. And so if we just make available 0.03 milligrams per kilogram nicotine on one lever or saline and nothing on the other, this is what we find. There's infusions plotted against a VR schedule, so variable ratio 3, on average, every third response is reinforced. You get nothing. I'm over here looking at Eric shaking his head, and he's like, of course, it's exactly what you get. This is Eric's and his colleagues' data who have studied this extensively and have defined a lot of the boundary conditions, of which nicotine, contingent nicotine, is not a robust primary reinforcer in and of on its own. So how can we solve this puzzle? Well, luckily, we've already heard somebody did it for us partially, right? So we've heard about Steve Goldberg figuring out what needs to be going on here. So we heard that that if nicotine functions as a reinforcer to domain tobacco smoking, it is likely that its reinforcing effects are magnified by interactions with interoceptive and exteroceptive stimuli, talking about the smoking and the taste and tactile stimuli, social settings, right? So these stimuli must occur together to maintain nicotine as a reinforcer. Here's just data pulled from his science paper. This is a squirrel monkey. You first see the nicotine plus the stimuli that we'll talk about a little bit in a different configuration. But you can see there's a lot of nicotine self-administration occurring in this monkey. As soon as you pull those away, you start to lose the intake of nicotine. As soon as you return those stimuli, the intake of nicotine is reinforcing. So yes, nicotine is a reinforcer when it synergizes with other stimuli. We talked about Dr. McNeely's data. As soon as she added a brief visual stimulus, a little different than what Eric's group used, but inspired by it, what you can see is the lower line is that the visual stimulus alone with the saline infusion. So the visual stimulus is a mild or weak reinforcer maintaining some lever pressing. That synergizes with the nicotine, and now nicotine is robustly taken along with the visual stimulus. And then this is just to really emphasize, put an exclamation point on it. As soon as Eric Donnie and his team give them the nicotine, what you see is, again, the visual stimulus maintains some behavior. The nicotine synergizes with that under these different schedules of reinforcement. Goldberg also gave us another idea of what was going on in this it's amazing how much was packed into this one paragraph in a science paper. He's basically talking about Pavlovian conditioning here. Thank you, Greg, for introducing Pavlovian conditioning. Really appreciate that. So he's talking about interoceptive and exteroceptive stimuli that are going to be in temporal contiguity with the reinforcing effects of nicotine. And if that's the case, then that these stimuli will become conditioned reinforcers. I will say this is the arrangement that he's talking about in kind of graphic terms. And so These are potential stimuli that might be occurring for a smoker or a vapor. So the taste of the smoke, the smell, all the kind of tactile stuff, the potential stimuli where you might be smoking, like bars, hookah places, your vehicle. Of course, now, of course, with all the vaping, the flavors and all those things are also there. Those are all to be entered into association with the robust effects of nicotine. Now, the puzzle for me, at least, is that we already know that nicotine's not a robust primary reinforcer. We learned that. So it's hard to imagine how those stimuli are becoming robust conditioned reinforcers. It's understandable how they're becoming conditioned stimuli for other effects of nicotine, but not a conditioned reinforcer for nicotine. And so I think for that, we have to sort of pause and reflect internally. And I mean that literally, reflect internally. So, okay, so this is now audience participation moment. How many people drink caffeinated beverages? Don't be shy. Yeah, all right. How many people have drank too many caffeinated beverages? Yeah, about the majority. Okay, anybody want to report to me what you feel? Panic attack. What does that involve? Heart rate. Sweating. Yeah. Jittery. Yeah. Right? Drugs have internal effects like caffeine that are stimulated that we can detect, we can report on, we can perceive. Charlie already showed you that it'll control animals' behaviors. And in fact, nicotine is no different. Nicotine has these internal interoceptive stimulus effects that are robust, detectable, and can control the animal's behavior. I'm going to propose to you that one piece of the equation that was not in Steve Goldberg's thing was that nicotine is also in a position to be a Pavlovian conditioned stimulus, and that it could enter into association with other reinforcers that are occurring in the environment. So this graphic shows that depiction. Namely, you have nicotine stimulus that's sitting around all of these great reinforcers, the lovely dinner we had last night, the tasty beverages, the social interactions that happen. Of course, those sweet flavors can get flipped as a reinforcer here in the vaping, and a huge, massive conditioned reinforcer these days is social media that could be in that position too, in that place. So the prediction then is that if you have the nicotine as the stimulus in this Pavlovian situation, it should be able to accrue conditioned reinforcing value on its own. So therefore, it becomes an amplified reinforcer through conditioning, through its learning history. Okay. Before I move on, I want to pause here to make sure I have time to just thank all of the people that have led up to this work and all of the thinking. And I have been absolutely blessed across my career to have amazing trainees and students along the way. So Joyce Bashir and Matt Palmatier helped with the early work of defining nicotine stimulus within a Pavlovian framework that we're talking about. Jen Murray came along and moved it into an intravenous model where the intravenous nicotine is now the conditioned stimulus. And then Sergey and Scott Barrett did the early work moving and testing whether, in fact, the fundamental nature of that nicotine was changed as a reinforcer by moving it into a self-administration task. And as you'll see, Dr. Barrett's been doing a lot of the work defining some of those boundary conditions upon which the nicotine has become an enhancer, amplified reinforcer. I know that was a spoiler alert. And then, of course, there's the amazing lab crews that have happened over the years. These are just two of the recent ones that have just done all of the amazing work and everything. So I just want to pause there and thank them. So with that, we have rats that are prepared with catheters, ready to start the experiment. And we're going to do the Pavlovian conditioning phase first. These are 2-hour sessions. There are 10 infusions per session. The rat doesn't have to work at all for any of these infusions. Here's the arrangement of the stimuli. So for the 2 critical groups to focus in on, the paired group, time is going along in the session, a 1-second infusion of nicotine occurs. 30 seconds later, they get 4-second access to liquid sucrose. Pavlov, bell, food. All you have here is nicotine, food is what's happening. It occurs very close in time. This happens 10 times across 2 hours. So the stimulus is not really nicotine. The stimulus really is the change in the detectable blood levels of nicotine that the animal perceives at the moment in time. The unpaired group gets the same amount of nicotine, gets the same amount of sucrose. We just separated by so far amount of time that we presume they will not learn about nicotine predicting sucrose. We presume until we run experiments, we don't know that. And so that's the unpaired group. That will be one of the controls we will use here. The measure of responding is how much they're going to search for the sucrose that comes in that dipper that the arrow is pointing to in that 30 seconds. So from the infusion to the sucrose delivery, we're going to look at that relative to the 30 seconds before the infusion. So a positive number is your conditioned response. It's your food seeking, your sucrose seeking, your goal tracking, depending on the words you would like to use for this. So here's conditioned response rate plotted by session. And what you can see is that if you looked at the lower level at 0, the unpaired group is hovering down there at 0, showing you no increase in dipper entry. So nicotine, just getting an infusion of nicotine does not increase putting your head in the dipper as the rat. And then saline, there looks like a little increase early, later on. Maybe they can hear some of the infusion pump or something. Curious, but not robust. What you can see is, though, that you get a robust conditioned response to the nicotine in that 30 seconds. So that increase in nicotine levels is detectable, functions as a stimulus, controlling your goal tracking, your conditioned response here. So we've established now that nicotine is got an association with the sucrose, with the food. Now the question is nicotine enhanced in its reinforcing value? Is it now a conditioned reinforcer on top of it? And so what we're going to do is move it to the self-administration task. And so for our self-administration task, I show you the configuration here. So you have 2 holes that are the nose pokes. One's active, one's inactive. We're on a progressive ratio schedule. We're not looking for breakpoints. That is our schedule of reinforcement. It's just a 2-hour progressive ratio. So for each reinforcer earned for each nicotine infusion, you got to do more behavior for the next infusion, then more behavior for the next infusion. So you got to work harder for each infusion. It's 2-hour sessions. No sucrose is available. The trough is still there, but there's no sucrose. There's no sucrose available in it. And we're going to do self-administration for 14 sessions. Here's the data. So you see active nose pokes. So this is your taking behavior plotted against the sessions, which again are 2 hours. You can see that saline is way down low, almost near 0. I should mention, there are no visual stimuli. This whole synergy stuff, we're keeping that away, right? I told you don't want those there if you want to measure reinforcing effects without the synergy of stimuli. So there are no lights or timeout stimuli involved here. So saline is low. There's nothing maintaining behavior there. So the unpaired group is showing you that in this situation, 0.03 milligrams per kilogram per infusion nicotine has some weak reinforcing effects in this condition here. So there is a bit of evidence for primary reinforcement under this situation. But what you can see clearly is that if there's repeated pairing of nicotine stimulus with sucrose, that the intake is amplified, and it is hugely amplified. We're looking at the output of behavior being more than doubled. So the Pavlovian conditioning of nicotine in association with the sucrose does amplify the intake of the nicotine as we would expect from a learning perspective. Just a little bit more data. I'm halfway. I could just add slides. I'm like flying. I need to slow down. That's great. This part of the experiment was a part of a larger experiment that we've not published yet. And so we know that temporal contiguity, how close in time the CS occurs to the US, so how close the bell food occurs, or how close the nicotine sucrose occurs, should matter. And so one way to manipulate that is by manipulating what we would call the interstimulus interval. So in this study, we're manipulating the interstimulus interval. So I showed you the 30-second group. And the reason I showed you the 30-second group is because that's the standard we've used when we were developing. When Dr. Murray started doing this work in their dissertation, only on the Pavlovian side, that was what we picked as our best guess. It worked, and we never did anything else until this study, where we decided to see where it would where the contiguity window would work for the infusion. So we tested 10, so we shortened the time between the infusion and the nicotine and the sucrose. We then lengthened it to either 60 seconds or 240 seconds, so the time between infusion. Now, if you notice the 240, if you go down and look at the unpaired graphic, the 240 is the sort of the short end of where our unpaired was. So we wanted to see if we had picked wisely in our unpaired group or not on that interstimulus interval. So basically the bottom line is for different groups of rats, the time between the infusion and when sucrose occurs is getting longer and longer. Where will they stop showing you a robust conditioned response? And does that parallel with the self-administration behavior or not? So here's the conditioned response data. So you've already seen the 30 group, which is panel B. Right? Now, if you just look back at the 10 group, you've got a bit more of a robust conditioned response with that shorter. I was a little surprised by this. We need to do another experiment now. I want to know what happens with like 5 and 1 and and Joe Ayers, my PhD mentor, would be really excited because I think we need to do a backward conditioning group here to see if that works with the. But yeah, that's crazy talk when I say it to my students in the class in my lab. The 60 seconds still showing you a little bit of conditioning, but much less robust. And the 240, you can't tell them from saline or anything like that. So 240 seems we're definitely on the outer end of where you can get control of nicotine stimulus controlling goal tracking the conditioned response. This is just a simplified sort of terminal performance. So just aggregating the last few sessions together so you can kind of see the pattern easier. So conditioned response plotted by the groups, unpaired is tagging the far end. Saline is on this end, and what you can see is 10 is producing the most robust conditioned response, and that's falling off as temporal contiguity is getting worse. Now, what happens in the self-administration? All the things are identical to the self-administration I showed you. They all go over in, they get the progressive ratio, 14 sessions, all 0.03 milligrams per kilogram, except for the saline group. And this is a little messy. I'll simplify it in a second for you. But what you can see is that the 10 group starts off strong, kind of ends up in the mess at the end there. Saline's way down. You've already seen the saline group, so they're not taking saline. And there is a temporal order to this. And so to stop the headaches there's the aggregate data. So it's easier to see the trend. And so what you can see is that temporal contiguity also matters for how robust of a conditioned reinforcing value the nicotine acquires, and hence how that translates into the amplified intake of nicotine as a result of the animal's learning history. So with that, there's no debate in this room, at least, maybe with anonymous reviewer, there is that nicotine is addictive is the major health issue. There's also debate on this. I just got whacked in a review on this one. But nicotine is a weak primary reinforcer if there's not the right environmental conditions for it to express its reinforcing effects. I'm really, really glad Jack's shaking his head yes right now, and I hope that yes is affirmative because I'm very scared right now. The synergy with the other stimuli, like Goldberg showed, like Eric Donnie and his team have shown, like we've shown, they synergize. You can see robust reinforcing effects of nicotine with the synergy of other environmental stimuli that are occurring. We're adding learning history to that complement, and that nicotine as a stimulus also is altered as a result of learning history. This is wildly important because if that learning history tends towards appetitive or positive or reinforcing, however word you prefer, you are going to get amplified intake of nicotine. I think the implications are very, very important and widely speaking because we've heard about importances of threshold, of dependence threshold, of reinforcing threshold, and learning history can alter the threshold by which nicotine may produce use, hence then misuse, and then dependence. And of course, this really matters to the FDA as they're starting and as they're thinking about, or not starting, they've been working on talking about what is the minimal value of nicotine in cigarettes and other tobacco products for this kind of threshold analysis. Just to think about how complicated this is, right? Because everybody in this room is different. Everybody comes in it from a very different learning experience. That's your smoker too, right? And so the implications for treatment and then are pretty complicated because you're thinking about their learning history. Less complicated for prevention, right? Because you can treat them differently if you can get out in front of it. So with that, I just want to thank again everybody who's been along with me in this journey. Is really, they are the inspiration. In the middle there is Dr. Barrett, who's a research professor in my lab, 3 grad students, and then Katie, as I've already introduced. With that, I've just really been grateful to be supported by, over the years, by the National Institute on Drug Abuse, NIGMS, Nebraska Department of Health, University of Nebraska, for this work and related work. And with that, I really appreciate you taking this journey with me on this path, and we'll take questions at the end of the panel. So thank you very much.
And next we have Dr. Caroline Cobb, who's going to be presenting on manipulating electronic nicotine delivery system characteristics to understand abuse liability in the clinical lab. And so Dr. Cobb is an associate professor in the Department of Psychology and director of the Health Psychology program at VCU, or Virginia Commonwealth University. Dr. Cobb's research interests include assessing patterns of tobacco use initiation, maintenance, and cessation, as well as refining of clinical trial and laboratory methods for product evaluation. Her areas of focus include menthol cigarette smoking and the use of other flavored tobacco products, including hookah, little cigars, and cigarillos, electronic cigarettes, and then looking at their effects at individual and population levels. So I'll turn it over to you.
All right. Thank you so much. It really is an honor to be a part of this meeting. I really appreciate the invitation and the opportunity to share some findings from our work with all of you. So first off, I just want to give an acknowledgement to essential contributions of my team members. Andrew Barnes is my behavioral economist buddy. He teaches me and keeps me in line on all the things. You all know Tom, I'm guessing from this room. Tom is on a boat right now, if you didn't know, sailing off into the sunset. We miss him at VCU very deeply. Also want to acknowledge Allison's contributions, as well as my team members, of course, all the staff that makes this possible, and the participants. These are my funding sources, and I have no conflicts of interest to report. But I do want to briefly state that my positionality as a white cisgender woman who also identifies as a former smoker, who's dedicated to addressing tobacco disparities, really does inform the interpretation of the findings that I'm going to share with you today. So let's talk about the goals for the talk. We're briefly going to go over ends, although I think most of you in this room are quite familiar with what that means, what that term encompasses, but let's just make sure we're all on the same page. Briefly talk about some clinical lab methods that I use to measure abuse liability, not all of them. And then we're going to do a case study on 2 particular ends characteristics that we used, we manipulated to try to understand their effects more broadly. And then I'll talk a little bit about next steps in this area. All right, so this is just a sampling of ends that I had in my office. There actually is one in there that's not nicotine containing. I'd be hard pressed for you to figure out which one it is. But as you know, these products, vapes, e-cigarettes, come in many shapes, sizes, and forms, which makes their study, quite challenging. But they do have some things in common, right? They all have a battery or power source. Some of those products allow you to change that battery or adjust the wattage, the power. They all have some sort of heating element, something that gets hot inside. Again, you can change that. You can change the the components, the size, the shape. And of course, we're going to find a liquid inside, and the liquid has to contain nicotine if we're talking about electronic nicotine delivery systems. But of course, you can use these systems to deliver other drugs if you choose. Now, I want to look a little bit more deeply at the liquid. I borrowed this slide from my colleagues at the Laboratory for Forensic Toxicology at VCU. This is actually from a study that we're doing where we're collecting confiscated vapes from children across the state of Virginia. So the schools send us the confiscated vapes, we characterize them, and then we actually look and see what's inside of them. So these are two products, Funky Republic and Raz. And this is an untargeted GCMS. And so what you can see are some of those components like nicotine in green. I know it's probably hard to see. We've got those active components. We have flavorants. We have things like vanilla. We have ethyl maltol, a sweet enhancer flavor. We have glycerin, right, that carrier agent. We have WS13, that synthetic cooling agent. We have menthol. We have a lot of different things, right? And of course, they don't look the same. It's going to be different depending on, even in the same batches of products, you'll see variations. And this also challenges the study of ends. So I want to talk real briefly also about how these products are regulated. As we know, since 2016, FDA has had the authority to regulate ends to protect public health. To date, right, they've authorized 39 products, which, that's unique products, but includes, roughly a few major brands. Lots of them have been denied. Now, the FDA has an opportunity to set product standards for ends, which they've yet to do, right? This could really be a lever for trying to help protect public health. And nicotine is, a likely choice, right? Nicotine-related standards could be a good choice because it's our primary driver of addiction. But of course, we're going to have to consider, right, balancing both risks for novel initiates by people who have never tried nicotine before, but also making sure products are palatable and promote substitution, right, from people using the most dangerous combustible tobacco products to move over and hopefully quit altogether. But of course, when we're thinking about a product standard for this, it gets complicated. And so I borrowed another slide. This is from one of my colleagues, Rose Bono, and I liked this slide because I thought it did a nice job of kind of thinking about some of the characteristics of ends and features about people who use ends that we have to consider if we were going to design a product standard around this characteristic. So right on the left-hand side, we have different things that influence, right, ends nicotine delivery capability. We've got the nicotine concentration in the liquid. We've also got the electrical power of the device. We've got the form of nicotine in the device, right? How much protonated nicotine versus unprotonated nicotine. Those things can influence the nicotine flux. Now, this is a term that my team likes to use. Not everybody does, but I like it because we can put all tobacco products on a flux standard. Flux is just the amount of nicotine that comes out of the mouth end of the product on a micrograms per second. That's all it is. How much nicotine is coming out. Now, of course, that micrograms per second, depending on how you use it, how long you inhale on a product, right, that's going to actually determine the dose or the milligram that gets ingested. But many other characteristics are going to eventually influence, right, the abuse liability, the abuse potential of that product. And of course, these things can also influence things like sensory characteristics. How does it feel when you inhale? And again, that's going to impact all these things in concert, as well as, of course, how fast that product is getting to the brain. So complicated. It's hard to control all these things. It's hard to study them, but we're going to try. We're going to try and see how it goes. So thinking about these methods, again, I borrowed a slide from a paper that I've been citing since I was in graduate school. I know some of the authors of this paper are in this room. It's one of my favorites. And so this depiction of abuse liability and thinking about traditional, right, well-validated measures and methods that we can use to understand abuse liability in a clinical laboratory context, right? We can look at pharmacokinetics, we can look at pharmacodynamics, reinforcing effects, cognitive effects, adverse effects, right? But it's hard to do all these at the same time or in the same place even. So let's talk about where I live and what measures I use. So thankfully, two of our prior graduate students who are now faculty and FDA staff let us take this picture, Dr. Sarah Maloney and Dr. Marjana Heiler, because we wanted to be able to showcase some of the methods that we typically use in the clinical lab with my team. So of course, we have a way to measure puffing behaviors, right? Puff topography instruments. We have ability to measure nicotine metabolites or nicotine delivery in the blood. We have physiological effects. We can measure those happening at baseline or in response to different doses, we can ask individuals to perform behavioral economic tasks, which I'm going to share some findings from those with you today, or just tell us how they feel. This is a great place where we can do a lot in this space. So now I'm getting into my case study. So like I mentioned, this study, or the set of studies, was focused on two of those characteristics that we think are essential for kind of, if we were going to design a product standard that says, hey, FDA, we think, you should consider these things in concert when you're looking at ends for whether or not they should be authorized for the protection of public health. Let's think about the nicotine flux of a product, as well as the allowable puff duration of that in that product. So potentially limiting the dose that the individual can receive. And so we've got two studies that I'm going to be showing you. Now, both studies use the exact same liquid. And I've got here the liquid concentrations as well as the flux concentrations. Those flux concentrations come from a mathematical model that was developed by our team members at the American University of Beirut. It's actually a freely available mathematical model. If you want to use it, I can help you. And so those are the flux calculations, and these are our four conditions. So we've got no flux, a product that has no nicotine in the liquid, what we're calling a low flux, around 36 micrograms per second of nicotine delivered, a cigarette-like flux around 90. And some people might want to argue with me about what's a cigarette-like flux. So there's an average of about 27 brands, again, from the same team, was around 80 micrograms per second. So we're close, we're in that range, around a cigarette-like delivery on that per second basis. And then we have our high flux, which again, you might be saying, well, that's high. That's pretty high. It's almost double the cigarette-like flux. And I'll say that perhaps wasn't completely intentional. We wanted to keep the power, the 30 watts, the same across all. These are all using the same device. And so we had some limitations here. And so again, probably if I had to go back and redo the study, I wouldn't use that high of a flux, but that is what it is. It's our high flux. So pretty high delivering product. Now, again, I mentioned the device. It was a tank-based device, same style of tank through all the studies, 30 watts and a 2-second puff duration. And I'll show you how we did that in a second. These are all the outcome measures, like the previous slide that I told you. These are the things that we measured in study 1, nicotine delivery, puff topography. Subjective effects in study 2 is going to be our subjective effects and our behavioral economic tasks. Now, here's how we limited the puff duration. So this was a device, right? You can't just go out and buy an e-cigarette or ends that has this capability. So we had to build it, or I shouldn't say I built it. The folks at American University of Beirut did. The first version was a little bit more clunky. You can see it's connected to a computer, a software system that allows us to actually set any puff duration that we wanted. And then our version 2, which was a little bit sleeker, a little bit slimmer, was what we ended up using for the majority of the study. You also might be wondering why 2 seconds. So again, this puff duration was chosen based on previous literature, right? Looking at both people who smoke cigarettes, right? So we know it's around 1.7 to roughly around 2 seconds on average. But we also did a study where we had people who smoked come in and use this exact same device with different nicotine-containing liquids, and they also took similar size puffs. On average, when people smoke ends, people who are current or frequent ends users, they usually take longer puffs for various reasons, but these studies were really designed to be focused on people who smoke. We're again thinking about the opportunity for ends to substitute or to change people's smoking behavior, so that was our target population. And I should say there already are ends on the market with puff duration limits, but they're much longer than 2 seconds. For example, Juul limits puff durations to around 6 seconds, as far as I know. And View Solo also has a puff duration, but that's not 2 seconds, it's much longer. So let's talk about study 1 and who was involved, both studies first. So both studies involved relatively healthy adults, 18 to 55, and they had to be current tobacco users, current users of cigarettes or ends. And you can see we actually had to adjust the criteria a little bit. Ideally, we wanted them both to be just people who smoke cigarettes primarily, but to make sure that we finished these studies and got to our recruitment goals, we had to make some adjustments. Study 1 included folks who could use cigarettes or ends daily, whereas study 2 was predominantly focused only on people who smoked at least 5 cigarettes a day. Now, all these liquids, these were custom liquids that we bought from a vendor. They were unflavored, unsweetened, basically propylene glycol, vegetable glycerin, nicotine, and benzoic acid. That's it. Nothing else, because we really did want to try to disentangle some of those other effects, those other factors from this nicotine dose delivery, which again creates different issues, because then you might say, well, then how is it representative of what people actually do in the real world? But again, another study. I think that's another study. So all these studies were within subjects, Latin square ordered. All the sessions had to be 2 days apart. And you can see study 1's 4 sessions and study 2 is 4 sessions, but we included an own brand control condition. Jumping into study 1 details, this study was recently published by one of our previous postdocs, Dr. Allison Petit. She's now faculty at ODU. 32 individuals, you can see from the slide, majority were current ENDS users, although we did have folks that were current smokers as well as used both products regularly. Before every session, they had to abstain for at least 12 hours. We did confirm that with blood levels after, but to make sure that hopefully also try to adjust for anybody who didn't comply, we made them sit for 1 hour in lab, supervised abstinence, before we did any outcome assessments. Now, once they got to the lab, what did they do? We did 1 directed puff session, so 10 puffs with 30-second IPI. Pretty common manipulation. We've been using this procedure for many years. With cigarettes, it's worked really well. I will say we're considering changing it, because if you've ever looked at naturalistic use patterns of people who vape or use these products, it's usually not 10 puffs in a row. So there's some potential issues with that design, but we've been having pretty good success with it so far. Then they had an ad lib period, and then at the very end of the study, we did an own brand challenge, which I'm not going to be talking about, but happy to talk about it later. So getting right into our plasma nicotine delivery data, you can see where the slide is set up with nanograms per ml here on the Y, and then our pre, post, and pre and post ad lib. And so right away, we're kind of seeing what we would expect, right? Those dose-related increases in plasma nicotine delivery by flux level. Remember, all the products, individuals were limited to 2 seconds, those 2 seconds puffs, and this is following 10, and this is ad lib. Not surprisingly, no flux and low, and sort of that cigarette flux right in the middle. But nobody's reaching what we might consider cigarette-like levels, like 20 nanograms per ml, for example, in that study, which is also not necessarily unexpected, given they might not have had experience with these products in the past. Now, subjective effects revealed similarity, I'd say, in both those dose-dependent effects, right? Folks were experienced some negative affect when they came in, because they shouldn't have been using any nicotine products. And so we saw that decline, again, the greatest decline with the high flux products, followed by cigarette flux, low, and no, in both the directed pout and the ad lib. But I want to show you some other subjective effects, which there's something a little different. So both individuals in the cigarette-like flux and the high flux level also reported some negative effects, right? These products, even though they were helping them, reduce that abstinence and craving, they were reporting high rates of sickness. They also had the highest throat hit and the highest harshness, so not particularly pleasant sensations. And what we saw again and then the positive one, so this this taste good revealed again lower levels, with the sort of in the between, the low flux being the most positively rated for taste good. So, right, this combination of both, relieving with maybe those negative symptoms, but also some negative sensations. When we look at puff topography, again, remember, puff duration is limited in the device. These are measurements of people's actual inhalation pattern. So just because the device turns off, right, the heater on the device turns off at 6 seconds, that does not mean that the individual moves the e-cigarette away from their mouth or stops inhaling. They could continue to inhale past that 2 second time. And what we saw is that people did do that on average, right? This is puff duration on average across all conditions. And we saw significantly shorter puff durations, even though, right, it was limited to 2 seconds, slightly shorter, because they were just pulling it away a little bit faster on average than these other conditions. And this resulted in less puff volume. So again, it's showing that people are sensitive to those potentially those subjective effects associated with those really high flux conditions. So now you might be wondering, okay, so what? Well, I'm going to show you now some more abuse liability outcomes. So that's just half the story. So let's talk about study 2. This was led by Rose Bono, recently published in Drug and Alcohol Dependence this past year. So again, 32 individuals. And I want to be clear, these are not the same individuals. They could not complete both studies. Folks, this was the same lab, but individuals had to be, these were exclusive groups. Now, in this case, remember, all are pretty regular, heavy, and actually pretty heavy smokers. On average, they smoked about 15 cigarettes per day for those who were involved in the study, with the majority smoking menthol. Remember, 4 ENDS conditions, Latin square ordered, but for every single person, the last condition was own brand. Again, before session, they were asked to abstain for 12 hours. In this study, we did not take blood, so we cannot determine if they did follow protocol, but we're gonna hope for the best. And also we did do we have some supervised abstinence that happens also in the study. Now, the procedures are quite different. Upon arrival, they're going to take 4 sample puffs of that condition-specific ENDS. This is their first experience with that condition-specific product, and then they sit for 1 hour. Going to sit with that effect. Then they're going to complete subjective and behavioral economic tasks. Now, I'm going to show, because everyone does this a little differently. I know this may not be exactly the way that some folks do this task, but this is our method. So we call this a drug purchase task. It's designed after the cigarette purchase task, right? We ask individuals how much would they consume, in this case, puffs, on a typical day. This is the hypothetical version, and the prices escalate, right, over up to about $20 in this case. Now, in this study, in this example, I'm really just focusing on demand intensity, although I do have the demand curves if you want to see them. But we're focusing on what people would purchase if the products were free by condition. So how many puffs, and we actually do the math for them using this survey, so as they can see how much money that these are going to cost. We also do a cross-product purchase task. In this case, right, where participants are reporting how many puffs of their own brand they would buy, as well as the session-specific N, and the price of own brand goes up, but the price of the session-specific Ns does not. And just like in the previous presentations we've seen today, we're looking to see, right, what do those Ns serve, right, as a complement, a substitute, or are they somewhat independent? I'll show you some evidence to support that. But the size and the magnitude of the coefficient really tells us, right, that answer. Now, lastly, we did a progressive ratio task. And I'll be honest, I'm a little bit scared to show this data because we designed this. We should have probably asked for more help, although I'm pretty sure I did ask Dr. Donnie at one point for some help. You did, you helped us. But we had never done this in our lab. This is the 3rd study we used the PRT. And every time we did it, we learned something new. And darn, I wish we had done it a little differently. But in this design, we had individuals working for key presses, right, for puffs. And they had, the first one was 10 presses after 1 puff, and the work requirement increased after 30%. And the task ended after 15 trials or 5 minutes of non-response. In this design. Remember, folks in this study, right, they've come in, they've had 4 sample puffs, they've sat for an hour, then they've done the purchase task, and this is what they do after the purchase task. So they've kind of had some time. Now, I'm jumping right into the results. Again, these aren't the pretty graphs. I'm showing you the coefficients in this case. But the first thing we're going to look at is that demand intensity outcome. And so circled in red are those coefficients. And so the main story is these products, again, people would, when they were free, they would purchase 150 puffs of their own brand. And when they're using any of the condition-specific ends, when they're free, they're not buying as many, which again, may not be that surprising. These products aren't serving as complete substitutes. They're not the most highly preferred of the folks as their own brand, but they're less than half in some cases, right? The high flux end is the lowest, 65 puffs on average, then when they're free, 78, 87, and the no flux end is the highest. Not quite what we expected. Now, looking at the cross product purchase task, right, we're looking at that, again, that coefficient, which is going to tell us whether or not these products substituted or not for own brand. And all the products were, the coefficient is positive. And so what that's telling us is all these, the ends product did serve as substitutes. So they were willing to purchase, as their own brand got more expensive, they were willing to purchase puffs of all of the ends conditions. And we did see a significant difference between the cigarette-like flux and the low flux, showing a higher preference for that low flux product, which again, wasn't really what we expected. We thought the cigarette flux product might be the most highly preferred in this study because we didn't have the results of study one. These studies were going on concurrently. Next, I'm going to show you data from the progressive ratio task. Again, looking at the average here, on average, people would work for 9 puffs of their own brand in this procedure. So willing to work for 9 puffs, and they're receiving those puffs in real time in the lab space. Now they're working from fewer puffs from all ends conditions, and we did see again that they would work for the lowest amount of puffs in the high flux ends. And with greater puffs for low flux and no flux. And this could reflect a couple of things. One of the critiques from the reviewers of this paper was, is it satiety? Is it the fact that, they're getting so much nicotine in those high flux ends that they don't need to take more puffs from these, that they don't need to take more puffs from that product? Perhaps that's what you're capturing. And it's hard for us to answer that question. We don't actually know, but at least we're able to say that yes, they were able to work a little bit more for more puffs. These are subjective data. I know this slide's got a lot going on, but some takeaways. What from this outcome measure in comparison to study one is we saw a lot of similarities. The high flux ends did reduce or were associated with lower rates of those withdrawal symptoms. They were associated with higher ratings of harshness, throat hit, not as pleasant. Low flux ends and no flux ends really did seem to be more highly preferred, at least in these terms of acceptability type measures. So in general, although the sig-like flux and high flux ends were, reducing those symptoms, or they were also giving some of these intense sensations, which also might have to do with the fact there was nothing else in that liquid to really blunt that, right? No flavorants, no menthol, no nothing. So just as a summary, cigarette-like flux and high flux ends, right, as I mentioned, did do a better job at relieving some of those symptoms, as well as having the most effective nicotine delivery, although still not reaching cigarette-like levels necessarily. So in this case, we determined, in this case, low flux end, at least under these conditions, did appear to be a better substitute for own brand, and people were more willing to work for it, more willing to choose it, to pay for it. So it's possible that maybe these high flux ends aren't really necessary. If we're trying to promote substitution for people who smoke, it's possible that we don't need these higher flux products. And one of the other outcomes from this work, right, I think there's still a lot of open questions this work opens up, but what I think this work also did was, it was the first time for a design or a study like this to really manipulate and control these two characteristics in combination. So it's a place for us to go next, or a place that we can continue to work on. And we have to, of course, start to think about other liquid characteristics, which also could be potential targets for product standards for the FDA. And so this is just a teaser. I'm not going to be showing you data from any of these projects, but this is coming to a journal near you soon, I hope. These are out for review, or about to be out for review. I have 2 graduate students that just recently defended their theses. This is Mary Carrico's work, where we manipulated the sweetener, or what we call a sweet additive, ethyl maltol concentrations and protonated nicotine concentrations, really similar design to study one. So we looked at nicotine delivery, subjective effects, cardiovascular effects, some exciting work. And I'll give you a little tidbit that this condition, the sweetened, not surprisingly, sweetened, highly protonated one, did seem to win out, particularly when it came to those positive subjective effects that we might be most worried about. Lizzie Ogunleya, she's another one of my graduate students, also recently just got her master's thesis, going on to a PhD. We were looking at some FDA authorized products. These are all Enjoy products. Some of them weren't authorized at the time we started the study, but now they are. And we were wanting to understand, right, whether flux, potentially other product characteristics, and flavor influence these same outcomes. So this study resembles more study 2. We did purchase tasks. We did not do a progressive ratio task in this design. We decided to take a break from that one. And also got some really nuanced, exciting results that really showcase how flavor and smoking, people's own brand smoking stat, or own brand menthol status, really impacts preferences for these products. So hopefully we'll be out soon. And with that, thank you so much for my time. These are my resources from my team. This project that I'm working on Virginia schools, as well as an opportunity, if you want to join us in Virginia or learn more about youth prevention happening in Virginia, you can join our coalition. So thank you.
have lots of time for questions. And so the presenters can come to take a seat up here, and then the microphone will be back there.
Oh, we're taking your
Is
time.
this ready now?
Extra.
You know, I would have given it to you if I knew I had extra.
Okay, what a fun session. Charles, I'd love to have a video of that. I give it to our clients because that's everything in a nutshell. By the way, FDA CSS staff, when you approach them, are great on interacting. And I think at least 4 or 5 times a year, we have meetings just to discuss alternative approaches. But so many sponsors just say, oh, but we've got to do the FR10. Anyway, that was really nice. Rick, you got that. I loved that presentation. And part of the magic of what Steve Goldberg did that you showed so nicely is he brought the cigarette key characteristics into the lab. And before him, John Dougherty, by the way, who was a Minnesota alum, he worked in that same lab I showed. He used different parameters. Nancy Ator at Hopkins took a shot at it with baboons. And Steve hit the right, very tiny doses, very quickly given, and with the stimulus. So there was also a duration effect. And if you think about it, it's more like a puff. So it was interesting, but when Surgeon General Coop found out, oh, it's addictive, and he looked at something and he went to the press and he said, it's 10 times more addictive than heroin. And so, of course, NIDA sent the press to me to explain what Dr. Coop meant, and I couldn't disagree with Dr. Coop, but I could say what Dr. Coop meant was at the public health level, there's a lot more damage, blah, blah, blah. But in talking to Dr. Coop, he could explain that there's a lot more to nicotine reinforcement, that the conditions under which it serves as a reinforcer are more limited than for cocaine and heroin in terms of laboratory parameters. And in cigarettes, it's perfect. But the other thing, there's 2 other things in cigarettes, and Victor Noble demonstrated this. Acetaldehyde is one of the substances that has an additive effect. And there are other substances in cigarette smoke. So the cigarette smoke is a cocktail. And that, of course, we don't study in the animals. But the big thing, in the 88 Surgeon General's report, we had an entire chapter, and the working title was Therapeutic and Useful Effects of Nicotine. In the end, after review, it was changed to other reasons that people smoke. But a powerful motivator of compulsive smoking that was clearer then, and I think still is clear, is benefits that people derive. And I remember reporters would talk to me and they'd say, yeah, I tried quit smoking, but then I couldn't write. And they said, I know there's risks, but I'm not a writer if I can't smoke. Or of course, women, especially gaining weight and going back. And that's something that we really have to take into account. And so when we were a number of us, including with John Hughes, talking about nicotine maintenance in more satisfying forms, the idea was really accepting what to us was the reality that there may be some people who are not going to function right, feel right, do right. And how do you do that? And Dr. Coop, one of my heroes, he was great. He's just very pragmatic, like he was with sex during AIDS. He said, don't have sex out of wedlock, but if you do, use a condom. And he said, same thing with young people. He said, it came to my attention but I can't get young people to stop having sex. I want them to live, so they should use condoms. He got crucified for that, by the way. But I think that's one of the things that we miss in the laboratory studies. But getting the right dose, conditions of nicotine right, how much abuse liability is too much? When we were first looking at ENDS, Tom Eisenberg called me and said, you can't call them nicotine delivery systems because they don't work. Because his first study showed, I said, Tom, they're going to get it right. This isn't rocket science. But the first ones, they didn't work very well. What is the right level? And I think FDA is on the same learning curve that a lot of us are. And so it took them a while to figure out that that convenient products, as they put it, that deliver satisfying amounts are the right level. We need performance standards for safety from FDA. I mean, that is crazy that we don't have them. I mean, I think we all agree on that. In terms of the nicotine abuse liability, as I stated, it's got to be high enough to substitute for a cigarette. What is the range? And within that, and Carolyn, what you showed is that with higher flux, people take smaller puffs. So right now, the question is, what range do you give them that a heavier smoker can get what they need and a lower smoker back off? And lastly, nicotine gum, when it was first being looked at for approval, they applied for approval for 2 and 4 milligram gum. And a key FDA official said, 4 milligram gum over my dead body. And indeed it was after he died. And a lot more data showing, I won't mention the name, I mean, rest in peace, John, but not John Hughes, this was a different person at FDA, but that basically 2 milligram for heavier smokers was placebo. They left out response cost. It's a lot of work to chew gum. And for a lot of people, the treatment, Maxine Stitzer really understood this. How do we generate more gum use per day? Because with more gum use per day, there was less weight gain in women and better outcomes. Or add 4 milligram gum and you will at least reach some people that you weren't reaching with 2 milligram. There's nothing simple about this stuff, but anyway, that was really fun and a really nice overview. I didn't make any comments you want.
So what's your question?
Yeah.
I just want to know, was that recording? I hope.
Yeah. It should.
So,
Oh, just tap. Is it on? Is it on? I was just going to say, I was there when that study, that study that you mentioned, one of the first ones we did with e-cigarettes, where we found they did not deliver nicotine. And I remember the hate mail that Tom got mostly from the community that said, you did it wrong. We are definitely getting nicotine. And so, we changed the methods. We ended up bringing in experienced vapors to come in and use their products. And then we were able to say yes, under those conditions. But yeah, it's complicated. And yeah, just like cigarettes, I know it's different complications.
Well, thank you for these talks. These are really interesting. Charles, I have a question for you about the lorcaserin story, because it was so incredibly disappointing with the preclinical data looking so promising. And, Steve Negus wrote that kind of post-mortem on the whole experience, kind of arguing that choice in preclinical models might have better predictive validity. I was curious what your thoughts are with that. He claimed that when you offered choice for self-administration, lorcaserin didn't work. In the human lab, similarly, lorcaserin didn't work for cocaine. And then, of course, it failed in that clinical trial. So I was just curious what your thoughts were about that.
Two things there, I guess, Meg. One is, I think choice is a perfectly fine procedure. We've used it usually to augment the simple self-administration experiment on the front end. I think it's much more insightful to give the animal an alternative. Again, it sort of gets into the magic bullet issue. If there's more than one thing to do in life, the value of drugs as reinforcers might be easier to manipulate. So I like that. The lorcaserin story was a dead duck, though, because of its failure in the clinical trial and because of the increased cancer risk. And it's a really messy molecule. I suspect that more selective serotonin 2C receptor agonists and maybe a closer look at the clinical data for the cancer risk, because I think some people questioned how robust that was, could still lead to some fruitful endeavors for the 2C story. But yeah, I think choice is a perfectly fine thing.
Failed in the choice model with his monkeys, and so it predicted the negative outcome, what I think he was, and it failed in a human lab study as well. When what was the choice about? So anyway, it was just it was a really interesting postmortem on that whole thing, because we all want predictability and how to better model it.
I don't think we have enough data yet to say whether choice has a reliably significant advantage over simple self-administration for predicting responses in humans. Need more data. Because typically a client does not come to us and say, do a choice study. They do a simple self-administration study. I want to ask a simple question with a simple fixed ratio, yes or no, thank you very much.
Great, simple panel. I liked all the talks and I learned a lot, but I want to publicly admit some ignorance. And Rick, this question is directed to you. Like intuitively, I think normally when I hear conditioning, I kind of get it. Even if something like reading a B.F. Skinner book and like something about morals, and he'll explain, well, if you thought about this way, and oh yeah, I never thought about it this way. But I can't get the responding conditioning explanation for nicotine maintaining self-administration. That like what I understood, I may not have understood it correctly, but if there's no stimulus change in the chamber, then they don't acquire it. But if you like turn a light on soon after they ingest the nicotine, then they will continue to self-administer at a high level. Like, how do you think about that intuitively? Because unless is it that like the darkness is aversive and the light like brightens their day? I'm not making light of it. I just can't get my head around it if I'm honest about it. Like I see at a practical level, okay, now you could try some potential medication for reducing nicotine use, but at a conditioning level, I don't get it.
So actually that part of it, the reward enhancement part, is not necessarily conditioning, Steve. You can have a piece of it. People have studied it a little bit, and we have an expert over there, so I might be stepping over my bounds a bit, but it turns on and off like a light switch. If it was conditioning, it would show you patterns of extinction, and it doesn't show you any of that. So we really kind of think of it, and I'm borrowing an analogy from my research professor, Scott Barrett. We think of it as an MSG effect, right? So the flavorant that you add in food to make it more yummy and delicious and robust and savory. So MSG itself is, it's not much. But as soon as you put it with a little bit of food, it turns savory, and that food is much more delicious. You eat a lot more. It's a lot more reinforcing. It has nothing to do with learning per se. It has to do with the synergies we presume in the central nervous system, although that's an area wide open for research. Nobody has really done any neuroscience on it, and only a little itty bitty bit on the behavioral pharmacology.
The light is the MSG?
Yeah, yeah. Well, the nicotine's the MSG, the light is the mild reinforcer. So out there, like the things I put in there, like, the work breaks, like the nice sunset, the alcohol, those things all can interact with the MSG, the nicotine. And so all those things are more reinforcing. And when they're in the environment together, they can synergize. And if you set it up right, they'll increase nicotine intake to places.
This-
And as Jack pointed out, the cigarette is the device that produces that synergy so nicely.
So this is really like Eric's and.
It's
Eric's
exactly
did
Eric's
that.
work.
It's,
Yep.
Just enhancing the reinforcing effects of a lot of
Yeah.
different stimuli in the
Mhm.
environment.
Yeah.
Lights for the rat, but sunshine for us, and that.
That was the first part. Yeah. The second half is that nicotine is a stimulus and it can become a conditioned reinforcer. And then you don't need those lights and you can produce lots of taking. Matter of fact, the experiments going on right now in the lab are literally going on today. Like,
Oh,
we're asking if they will synergize, the reward enhancement effect will synergize with the Pavlovian conditioning effect to produce super amplification or not.
Okay, okay. Well, that helps. Thanks.
Yeah, you're welcome. It's a good question.
Not to forget that we use stimulus lights with the things that are already very effective positive reinforcers for a reason. You don't have to flash a yellow light every time a monkey gets an infusion of cocaine or fentanyl. They'll keep working for it. But if you do, you're going to have much more robust and lingering behavior. And you can attest to the value of those visual stimuli by virtue of reinstatement procedures. Just give them the lights back and they become very good reinforcers themselves.
And I intuitively, I did. It is, yeah. For some reason that's clear to me when you have a clear reinforcer and you pair the light with it. I don't have any trouble with that.
And I think,
Enhancing, like, okay, the MSG is the closest I've ever heard, you know, so the way you describe it is, it gave me some clarity. I still don't quite get how this keeps people who know, like, I continue to, I've got lung cancer, but I can't stop doing this thing. And, you know, it's so powerful, but maybe it's because there's so many stimuli in the environment that have been impacted that, and then, Rick, I think your model of then remembering the drug itself can now become the condition stimulus. So, yeah. I understand better. I have to admit, I'm still a little bit challenged at these lunatic level. And Charles, I understand the Operant stuff much better. And yeah, so it just is hard for me to understand how that produces the human phenomenon. Where you see in these hospitals, people self-administering nicotine, they under, like, yeah, dire circumstances. It's that powerful.
Great. My name is Xiao Zhongwen. I'm from University of Buffalo. I have two questions, one for Rick, one for Caroline. The question for Rick is actually linked to Dr. Steve's comment. So in this case, we have a lot of smoking relapse among postpartum women, like up to like age 80 or 90%, like 12 months after birth. You can imagine those mothers spend a lot of effort to quit smoking during pregnancy, stay not abstinent for like 9 or 10 months, but then after baby is born, you know, most of them go back. So if your studies show like you need a stimulus to keep using nicotine or smoking, how can we use that as an intervention strategy to prevent postpartum smoking relapse?
can I take the Greg deferral to think about it for a minute? That's an amazing question. I'm really gonna have to, can we chat just a little bit afterward? I mean,
Sure, yeah.
that's amazing. Yeah, let me think about that.
Great, thank you. My second question is for Caroline. So we know currently e-cigarettes are not approved by FDA as a cessation aid. However, a lot of researchers are trying to do that. So a common idea is that we maybe help people who currently use cigarettes, combustible cigarettes exclusively, then switch to do use of cigarettes and e-cigarettes, then quit remove the cigarette completely, then stay with the e-cigarettes, that eventually they quit the e-cigarettes, so become nicotine free. So it's kind of a stepwise approach. So then your studies show like it's like between the high and lower flags. I got a bit confused. Like you said, like in terms like the pleasure or satisfaction, a lot of smokers actually, your test showed that the high flex e-cigarettes give people similar feeling as a cigarettes, right? Then later you have another a study show like we don't need to get a high, low maybe even works better, whatever. So in this case, I got confused, like which product should we use if we want to help exclusive cigarette users to switch to dual use, then eventually to quitters, both products?
That's a great question. And it's funny, I was actually thinking about another paper I published, a study that's a little bit better designed to answer your question. So, these studies weren't necessarily switching studies, right? We're kind of evaluating these abuse liability indices. But I did do a study where we did ask people to use e-cigarettes that varied in their nicotine concentration for 6 months. So people who smoked tried to substitute and reduce their smoking for 6 months. And in that study, we didn't have a high flux. We had what we called like a cigarette-like flux, a low and a no. And the best condition, the condition that worked the best for folks was the higher one, the highest, the cigarette-like one. People were able to substitute to the greatest extent, reduce their smoking to the greatest extent, reduce their toxicant exposure to the greatest extent. So I was thinking about the study when I was writing up these conclusions, right? Our clinical laboratory designs may not, again, this condition was not the same, different e-cigarette, different product. But I'm going to get back to your question. What's your recommendation? What do you want to do? I mean, I think what you described is exactly what I would suggest, right? Dual using as if you have to, if you can completely switch right away, ideal, and then obviously get down completely off of e-cigarettes or ends as soon as possible. But I do think that people need to use a product that is going to relieve their cravings and withdrawal, right? It's not going to be an effect. You're not going to be able to substitute if you're not able to reduce those feelings, right? That's why nicotine replacement is so effective, right? The question is how much nicotine is necessary, how much flex, how much nicotine delivery. And so what we found was that those high flex products, while they were effective at delivering nicotine and reducing some withdrawal symptoms, they weren't necessarily the most pleasant to use. If they're not pleasant to use, people won't use them either. So you got to have a balance of both, acceptable, but maybe it doesn't have to be as high nicotine. So maybe somewhere in the middle, that sweet spot where it is, we don't know yet.
Great. I would have thought about that because a lot of my patients complain, saying that the e-cigarettes don't give them same level of satisfaction as the cigarettes.
Yeah.
But when I ask them, what can you explain? What's there? They cannot tell the specific reasons. Like when I ask about the nicotine concentration, they say no, exactly.
Yeah, I think it's not the same, right? The aerosol that comes out of an e-cigarette is not, certainly no shape or form similar to a cigarette smoke. And so I think those sensations, those experiences, they're very different. And people are looking for those conditioned, right? Those conditioned stimuli, and they're not the same. And we can't make them. I mean, maybe the industry can, I don't know.
Just say like they keep using the word of weaker, but I'm not sure that weaker is nicotine association or other thing else. Like those things kind of make the respiratory system like you stimulate that, that noise doesn't exactly addicting.
Yeah. No, I've heard very similar from our participants. We interview them and say, how's it going with your e-cigarette if they're trying to reduce, and I hear a lot of the same. It's not the same.
Right.
Here it's.
Thank you.
Charles, I hate to bring up Lacasterin again, if I'm going to bring it up, maybe for a good reason. So the clinical trial, the placebo group had less than 5% of people meet the clinical endpoint of cocaine abstinence at 13-week endpoint. And so, and I bring that up because we talked about that study. It's true. If I think a lot of these randomized clinical trials we conduct with the medications developed preclinically through phase 1 testing into these large-scale clinical trials. Substance use disorder is really hard to treat. It's really hard to maintain abstinence, but less than 5% with the sort of baseline of what we would expect in a placebo condition is sort of abysmally low. And so I think it sort of speaks to, and we're trying to pit these medications in traditional clinical trials. And you said something in your talk that sort of reinforced that point. We're probably not testing them under the ideal conditions to actually see a medication that may be helpful for a subset of patients or helpful for all patients to at least a little degree. And so the question kind of for you and for everyone else and anyone else for that matter is, how do we do that better? I think you have the vantage point of seeing a lot of medications come through preclinical settings that truly, I think if we had tested in the right way, would be on the market now and be helpful. I think we've talked a lot about reduction as an endpoint, which is a great one. But I'm curious if there's other thoughts of either endpoints or how we should be conducting these studies to better identify those signals that may get them through the FDA, that may make them available. That's $1 million question.
It's not really a question for me. It's a question for clinical people because I have always been suspect of the rigor with which those experiments are conducted. My monkeys are 100% compliant.
Mhm.
And I know that's not true when you move to the next level, and it's much harder to do in humans. I appreciate that, and I have no doubt that we have looked at things which did not advance because they failed in clinical trials, just because the clinical trials were not run properly. But there are other people in this room who could speak better to that point.
And I guess maybe not to make you say it, but or to make you delve into it, but sort of what do you see as the biggest gaps in that manner? So you can talk about non-compliance in terms of medication adherence. You could talk of fidelity of behavioral intervention. You could talk about all those sorts of things. I guess you see something that's been, at least from your vantage point, if we're going to put priority to improving the rigor of how we're conducting it, recognizing it's harder, it's easier said than done. There's a lot of things like, do you see something that's been the biggest gap? If that makes sense, not to put you on the spot on that, but I see Sandy standing up too, who may have her own thoughts.
Yeah, let Sandy pipe in here.
Yeah, just because I was about to ask you a similar question, but from the other direction. I think one of the big gaps that's missing actually in the connection between animal and human research is the lack of attention that's paid to adverse effects of the medication, right? So in a lot of the animal studies that you run, I mean, you were on my dissertation committee and you taught me that, you know, you push a medication or a dose of a drug up until the point where there's no more responding, right? Which is fine. It makes sense from a pharmacological perspective. But you know, for those of us who have made the jump to clinical research, we can't do that for a variety of reasons. Sometimes it's a safety concern. You know, we can't test the high doses of lorcaserin that you tested and show those really beautiful reductions in, you know, self-administration behavior. So I was going to ask, is there any way that you can improve the predictive validity of the animal models by focusing more on adverse effects or dose limiting things in humans?
Well, that's a good question. And I would argue that for our non-pharma sponsored work, we do that. We tend to push things as far as we can because that's the way we've been trained. But for 99% of the things I have done from people in the pharmaceutical industry, it is X-fold of our expected clinical dose, no further, or I'll kill you. They don't want to know the answer at that end of the spectrum. And you know, I used to say, well, gee, but you know, people I know who treat people who have substance use, these people will take massive doses. Shouldn't we really study those doses? The answer is always no. So I don't know how that disconnect can be bridged, depending upon who's paying the bill. It's a very narrow spectrum that they want us to investigate with regard to new chemical entities and their abuse liability. And they're not interested in adverse effects. By that point, they have done their safety pharmacology and toxicology. They know what's out there from an animal's perspective in terms of basic physiology, maybe not in terms of things that would be more relevant to abuse liability.
I mean, I think it would be helpful if there was just, you know, some sort of, and I don't know, maybe it's not possible to do this in the animal state. So this, like, if we focus just on a medication potential impact on a drug of abuse, right? You know, there are data in the animal toxicity studies, for example, that say, okay, you have to stop at this dose because you're seeing this cardiovascular signal, right? So that means that will be dose limiting in people. So like in the lorcaserin study that we did, in combination with oxycodone, we couldn't go higher to higher doses of lorcaserin because of potential toxic effects because of, other things. And so there was no effect, in that situation. And, maybe you know more, the clinical studies that were done with it in the bigger treatment trials, they had the same limitation. So, it's essentially in the animal model, it's a false positive for the medication because you're exceeding the safe dose limit of the medication.
Well, that's false positive in quotation marks.
Yeah.
Were you able to run those doses higher, you might be able to see the same effect. And we recognized that at the time. And in fact, we subsequently put in a grant and got it to combine lorcaserin with buspirone, with the thought that we have two drugs which have a hint of a signal in various places. If we put them together at smaller doses, we might get a good therapeutic effect. And we got some very good data with that, although Nora Volkoff didn't think that we could achieve the appropriate clinical doses, but in fact we could in animals. And that would have allowed for, I think, reasonable studies in humans, but the plug was pulled on lorcaserin at that moment. So those drug mixture studies also terminated, but they were far more promising than lorcaserin by itself.
I would just add to this conversation. I've speculated a little about this when I was doing some vaccine work, vaccines against nicotine and meth and things. And our rats, quote unquote, are normal. They don't have comorbidities and they're not a messy population like those people who are using cocaine, nicotine, and those things. And I have always wondered and thought it would be fun, and if there was unlimited resources, to use those kind of messier animal models in a more clinical trial setting. But of course, that you then lose some of the advantage of the animal model. I can give you my address and my Venmo account.
Go fund me lot, please.
This is back on, well, two comments, but the first was on the MSG. The last time I read about pyrazines, they hadn't figured out why pyrazines were so effective in promoting nicotine addiction. But that may be out of date. I've not reread on that. But I'm wondering whether, since the tobacco was always being delivered up with pyrazines at the same time that the cigarette was being consumed, whether that could be like this other, stimulus, that was just speculation on my part, and I didn't know if anybody had looked at that. So
I wish I had my notebook up here right now to write those things down. Absolutely. I think the comorbidity, I think Jackie, you referred to another compound that occurs in nicotine or in tobacco that are constituents that could all interact. There was a lot of interest in these constituents and how they interact with nicotine. We have not touched any of that in these models, and those would be a fantastic path of research to know how they interact, change the stimulus, don't change the stimulus, enhance. Yeah, it would be amazing to do that.
I believe it's Brian Henderson at Marshall University who did the work on that. Yeah, if you're already familiar with it.
Yeah, it's beautiful work. Yeah.
The other question was kind of similar to something I said earlier in terms of dual use, that the actual title on the American Cancer Society page, for those who aren't familiar, is smoking and vaping together increases lung cancer risk more than smoking alone. And the text says, in fact, people with lung cancer were 8 times more likely to both smoke and vape than people without lung cancer. That's just a part of the paper, but even skipping aside the acute effects, just focusing on the lung cancer, it's still very hard for me to understand how dual use, even if you're decreasing number of cigarettes smoked with this type of, we're getting into epidemiology here now, not just like theoretical toxicology is what the industry relies on, right? We've gotten to the level of epidemiology, and it's just still very difficult for me to arrive at a place where dual use is, because it was Hayek's study, actually, interestingly enough, the most cited one, which he created more dual users than successful quitters. And unlike the people who are on NRT in Hayek's study, they were continuing to use many long after they were still using vapes. So anyways, I'll drop that as a question and yield the microphone.
Well, thanks for bringing that up. I think that I was a graduate student when we first started studying e-cigarettes and knew very little about them at the time. And I really have, pride myself that I continue to just be open to the science and follow where it goes. So my opinions have changed. I mean, the past 20 years, right, from being very skeptical to be very positive, to being more skeptical, to being less, going back and forth. And I think that's the real key for scientists to keep that mindset. And unfortunately, I don't feel like a lot of folks do. They have a mindset, they have they potentially read a few articles or have this mindset towards a certain set of products and refuse to see the other side. So thank you for bringing that up. And, there are certainly studies, I've just reflected on my own work, studies that I would not perform today. Knowing what I know about e-cigarettes, for example, there are certain studies I would not have done or I would have not have proposed. Those changes, I mean, that's what we do. We improve, right?
Absolutely.
So.
In the old days, we could categorize drugs really simply, stimulants, sedatives, and opioids, and they were pretty robust. And then there were, cannabinoids and psychedelics that are more variable. But what we're seeing at Pinney Associates is a lot more drugs that are novel acting, relatively low abuse potential, and a number of them, you push the dose and you get negative effects. And that's being considered by FDA, and we've dealt with that. Now, I can't talk about which drugs, and some of them are approved on the basis and scheduled on the basis. They could have been scheduled more restrictively, but they're scheduled less restrictively because FDA recognized that their actual real-world abuse potential is limited by the fact that the effects start getting aversive as they get higher, which is interesting. Nicotine in some ways is a good model for a lot of that. And one of the things that I'm not sure if you want to call it, I call it malleable stimulus or variable. But one of the things that Goldberg and I found in animals and humans is that the same dose could be a positive or a negative reinforcer. And one of the really cool studies that we were doing when my lab was shut down by the White House, the nicotine part of it, was that people could press the lever to self-administer 1.5 milligrams of nicotine. But I think it was every 30 minutes, 1.5 milligrams of nicotine was a programmed infusion, and they could avoid it. So they had two levers, and guess what they did? They avoided the program, and they self-administered on their own time. I think 2 subjects of data are published, but then the study was stopped, so it never was completed, but it's in Pharmacology, Biochemistry, and Behavior. I think part of the magic of the cigarette, Mike Russell said it, a couple of decades ago, fingertip dose control. And part of enabling people to get what they need or want, the substitute for a cigarette, is coming closer to that. Gum doesn't do it. Patch definitely doesn't do it. All of those products I'm proud of, I helped develop with them, and they help a lot of people. But it is really hard to beat a cigarette in dose control. And people vary widely, not only across people, but within the day. What do they need? When do they want it? How much do they want it? And again, 2 other things going to Steve Higgins' point in question, how can this be so strong? First, again, it's not a cigarette is not a single entity system. Right. And we see that with kratom or kratom, which is a symphony. It's lots of alkaloids. And for a lot of people, taking mitragynine out, that's kind of like it's caffeine, is good enough. But a lot of people say, no, I need kratom. Now, this thing I mentioned this morning is a whole different animal. That's 7-hydroxy. But so the cigarette is a cocktail. And the other thing is the benefits. And if I've got a morning where I've got to get a lot of writing done, I had a shot of espresso to my coffee. The useful effects, the effects that people want or need, and this became real to me in the 80s at NIDA when we were working for the Army, Air Force, Navy, Military Performance Command, and they had a practical problem. F-14, and F-15 pilots were taking their respirators off and chewing tobacco. And the Navy Surgeon General said they claimed they would go into withdrawal. And he said, we don't have any tolerance for problems. So we used the military cognitive performance battery to look at nicotine withdrawal. Said, yeah, within about 2 hours, they're making more errors. They're slower to react. And so the military was really pragmatic. It was really cool. They lifted the ban on gum as pertained to nicotine gum on an aircraft carrier, Delano Roosevelt, going into the Middle East in 1982. And they said, we want the troops to quit smoking, but most of them aren't. So how do we maintain performance? Same thing, by the way, happened with the FAA when they very carefully took smoke out of the cockpit. And me and Mike Fiore and some others did the report on how to do that and minimize the risk of airline crashes. There are some real practical issues, but those daily benefits, we don't like to talk about the benefits of nicotine. And in the 88 report, we didn't use that word. But addiction is complicated, and all addictions are complicated. A lot of people get, when I went to the Addiction Research Center, Don Jasinski and others said, look, a lot of the quote addicts talk about, this is the only thing that got me on the norm. There's nothing simple about this stuff.
I was reflecting more on, what was my, what's my concern? And I think I'm still wondering about the validity of the animal nicotine self-administration model. But I say it respectfully, and I'm willing to be, corrected and educated. But like, I'm thinking about cocaine, which, I devoted years of my life to and I just found so compelling, like the studies by Crissell and Johansson and Balster, like you allow those primates to have the non-human primates free access to cocaine, they'll just kill themselves. And it just seems so consistent with what we see out in the streets and whatnot. So are there examples of like rats who have been taught to self-administer nicotine where they'll persist, or the behavior, you get like this kind of behavior that mimics what we see in like the cancer patients who just can't quit, even though they know that it's killing them soon, in the near future. So yeah, I throw that out and want to hear what you guys think.
No, you. The nicotine guy.
Squirrel. That would be a great model to work. I mean, there's certainly persistence in behavior. You can do abstinence periods and it relapses, but none of our animal models really capitulate that with nicotine to that extent. The way I think about it, Steve, and hopefully maybe this will help a little with the struggle bus, is that any one piece of the animal model, at least the way we approach it, is we're isolating a factor and bringing all of our possible control to that factor, right? So I'm getting rid of the Eric Donnie light in my model because that's not going to help me interpret nicotine as a conditioned stimulus acquiring condition reinforcing. It'll confuse that piece. But all of these things we've heard today so far, and we're going to continue to hear, I think are all factors playing a role in individuals. And I would add that not every factor is equal, and not every factor is equally weighted in individuals, and that there are different factors for different people across different places, which why it makes this such a hard health concern. And so those individuals you're talking about, to recapitulate those models of those individuals who have spent their life smoking, who my grandmother was one of those. I held her hand every night when I came home from my job at Pizza Inn. 2 o'clock in the morning, she'd be up, in pain from cancer, dying slowly, still smoking. I get it. And that's more than just the little snippet of the learning I'm studying right now. It's more than the snippet of this. It's all of it combined in some way unique to her. How did my mother then quit 2-pack a day smoking after she saw my grandmother die, her mother die from that, right? I mean, if we could figure all and harness all of that, we'd be further along than we are, but it's such a hard puzzle to piece together. And while I have the microphone, let's talk with Mark Boughton about renewal, pregnant women, and relapse, because I think that has some important pieces to it.
I know that we have to go. I just have one last question, and this might have to help to address Steve's point. I just wanted to begin by saying that I'm so grateful for the images of John and Warren at the beginning of this. They're missed dearly, and I appreciate that opportunity to reflect on their careers. But I can hear both of them saying, if we want to understand substance dependence, we have to understand both abuse liability and relapse and what happens during discontinuation. So I guess one question for you, Rick, you have beautiful data, always gorgeous. How do these interoceptive stimuli that are associated with nicotine, how do they function during discontinuation and withdrawal? When we think about the full lifespan, the life cycle of somebody who learns to become dependent upon nicotine and then they try to unlearn it. So if you just talk about that for a minute. I know maybe this is a softball, maybe it's unanswerable.
No, it's not a softball. It's a great question. You'll never unlearn it, as we know, right? It'll always be there. Can we develop competing learning to help encourage the behaviors we want, like contingency management provides competing behaviors and competing learning history. We should sit down and write the next grant. That is actually the next grant to isolate some factors that are into those. The one thing we do have written in the grant right now that we're going to be mucking around with is, and I'm going to use jargon, I won't explain it, but I'm happy to explain it, is over expectation. We have built in to see if we can make use of that in a way that might decrease. One of my favorite comments from a reviewer, besides the one I showed you, was it's like, last time I checked, the National Institute of Drug Abuse is also like trying to help cure people, not like increase their liability. Do you have anything in your models that could decrease? I've taken it to heart. So every time I write a grant now, I actually build in an aim that tries to reverse engineer things we're doing. Over expectations, the reverse engineering this time, but there's a lot more reverse engineering, as you pointed out.
All right. So one apology to Carolyn for obviously messing up her progressive ratio. And for a little bit, just a comment on the predictive validity issue and the translational issues, a little bit to Sandy's questions as well. One thing we're not talking about that I feel like we miss a lot is an old concept. It's one that's been around a long time for behavioral economics, and that's the concept of utility. That we often talk about these things as if they're constants within a person over time and condition. And that's, if you work with patients at all, that's not true, that at an individual level, the utility of a drug varies, and over time and condition, it varies. And so part of, I think, what we are missing is the ability to think about reinforcement while we also think about the utility and why it's functioning as a reinforcer and how to embed that more in our assessments. So just a challenge for us to do more in that space and think about the why a little bit more, that Jack and others have pointed out as well. There are reasons people choose to do things that relate to their predicted outcomes. And those predicted outcomes are relevant in some conditions and not others.
Thank you so much for that. And I want to say no apology is needed. Just one more thing while I have the mic. I meant to mention it while I was up there, but I really want to give a call out and remembrance of Warren Bickle's contributions to the work that I presented. I meant to say something, but I got a deer in headlights up there and didn't do it. But he was really, really consequential in the work, all the behavioral economics works that VCU did. He was a consultant, a collaborator, constant, sometimes a critic when we needed him to be, and just really, really miss him and thankful.
Do we still have time? I just want to make sure, because I do have a question for Charlie, and I don't know if I'm allowed to ask a question of the panelist.
Let's give the audience a bit of speak.